Thanks, Mike, and I'm clearly the sidekick to Roger, so I'm happy to be here. Thanks for the kind introduction. So what we're going to talk about is how does this biology play in the clinic, at least from some experience that we've had. And it starts by understanding that this is a cancer that in 50% of the cases is driven by a single nucleotide change that changes one amino acid that takes a protein that should be on or off to be on constitutively, and then it tells the cell to be on cell cycle all the time. As we heard from Mike's excellent presentation this morning, several other things need to happen, but this continues to be the driver that tells this cell, you're a cancer cell. There has to be dysregulation here, there has to be avoidance of apoptosis, avoidance of the immune system, as we'll hear this afternoon, but this continues to be the driver. So if we simplify biology this way, then we can think, if we block the driver, then should we have responses, and then should we have these cells stop growing? And actually, I'm not going to go back on the data, I'm not going to go back 4 years. We all know this. If we select patients based on the presence of the BRAF mutation, and we give them either vemurafenib or dabrafenib, two inhibitors that work very well in blocking this activated enzyme, then we get this waterfall plot of responses where very few patients are naturally resistant. The great majority of patients have different degrees of responses, something that it's worth studying, but it tells us the biology is right, that this is a cancer driven by this mutation. But as we know, the problem is that as you continue giving the drug, that in many cases, not all of the cases, but in the majority, the tumor will start regrowing. I'm following patients from the phase 1 trial of vemurafenib who are now going for 4 years and counting, and they continue to respond. So don't let anybody fool you by saying that it only works for 7 months, because that's what happens to patient number 50 in a group of 100, and the patient that you're seeing may not be patient number 50 in a group of 100. But once this happens, then can we continue to simplify biology and try to understand it well? And I'm lucky to be working with Roger Lowe on this to be able to understand things, but I think I've taken some slides out of what I wanted to say, but here is where we have understood that as the tumor becomes resistant, and Roger will tell us more afterwards, in many cases, it reactivates this pathway, at least in Roger's experience, probably 2/3 to 3/4 of the cases. It induces a series of alterations that I'll leave to Roger to explain that allow the reactivation of this pathway. That has led to the thinking that if the pathway is turned on again, then can we block it at another level and then get a secondary response? The mechanisms of these are BRAF amplifications that Roger described, BRAF splice variants that Dave Solid described, secondary NRAS mutations, also by Roger, a series of other alterations by signaling molecules that can fit into here, or secondary mutations in MEK that can also reactivate these, most of which should be targeted by blocking MEK afterwards. So if this is the understanding of the biology, then how does this play in the clinic? And here we have 3 of the patients that Roger and I have been treating. These are patients who were on vemurafenib, and at some point after being on single-agent vemurafenib, they progressed. And at that time, Roger does a lot of biopsies, and we order a lot of biopsies and trying to understand what happened in this tumor that's different from a baseline. And then these biopsies are taken to Roger's lab, and Roger, after a week or a week and a half or 2, sends me an email or pages me and says, this patient has an amplification of BRAF, and I predict that this patient will respond to adding a MEK inhibitor. This patient has a splicing of BRAF of some exons that allow the protein to overactivate by homodimerizing, or this one has another BRAF amplification. So we understand this, and then we have a clinical trial that would allow the patient to continue on the BRAF inhibitor, and then we add a MEK inhibitor because we think that that would take care of this new signaling through the pathway. Then we can have something like this. These are the same 3 patients with a PET scan 50 days after adding a MEK inhibitor it was called GDC0973. Now it's called cobimetinib. You can see the night and day difference in all of these patients. This patient had biopsied this lesion, but this lesion responded and many others responded, but this one progressed, which will go to something that I think Roger is going to tell us about, which is that once we try to chase resistance, we can find tumors that have found several ways of progressing. But this is the proof of concept that it's there, that we can treat resistance. It's not too good at treating resistance. It doesn't last that long. But if we try to then take this in an upfront setting, and that's been done by the combination of the dabrafenib and trametinib and published by Keith Flaherty and others, that you can actually get better responses and more durable responses. As we do this combination, there was something that was interesting, which is it would also address another problem with single-agent BRAF inhibitors, which is the development of secondary squamous cell carcinomas. And I'm not going to go back in time. Most of you have heard this biology several times about paradoxical MAP kinase activation. I'm just going back on the data so we can then understand how can it play in the clinic. So here we have, we put together data from several centers with investigators from Roche and Richard Moraes and our group, and we put together the squamous cell carcinomas. We thought that it would be from paradoxical activation, and that would work by having an upstream mutation in RAS that would turn on the pathway as we gave the BRAF inhibitor. And you can see these cells, these colonies progress instead of responding once we give the BRAF inhibitor. That's because they have a RAS mutation on top. It changes the signaling and actually gives the opposite signaling than melanoma when we give the BRAF inhibitor to squamous cell carcinomas that have a RAS mutation. And Richard Moraes set up an animal model where he induced squamous cell carcinomas by giving 2 carcinogens. If you gave the BRAF inhibitor, the squamous cell carcinomas appeared more rapidly. It didn't appear more of them, it just shifted the curve. The BRAF inhibitor is not a carcinogen. It has no feature of it because this happens within days of giving the drug. If you omit the carcinogens, it doesn't happen. So it's from activation of the pathway as opposed to being a secondary malignancy. But I'm showing this not to go back on all data, but to say that Richard also had an interesting group here, which is he gave the BRAF inhibitor plus the MEK inhibitor, and then all of these squamous cell carcinomas did not appear. So that's great if we're trying to treat squamous cell carcinomas in the mouse, but does it work in the human? Well, this is a patient that Roger and I saw probably a couple of years ago that came being on BRAF inhibitor alone, had a whole bunch of squamous cell carcinomas, and too many to be treated. So that, if we go back to the pathway and we understand this phenomenon of the paradoxical MAP kinase activation, where we give the BRAF inhibitor, it transactivates CRAF, and it increases signaling. The thought would be that if we give a MEK inhibitor, then we should prevent this paradoxical activation, and we would decrease the pathway signaling, not only in melanoma cells, but also in the squamous cell carcinomas that are developing. And this, what we did with this patient was on the BRAF inhibitor, we added the MEK inhibitor, and you can see this night and day difference within a short period of time of developing these secondary squamous cell carcinomas. This is data that's been generated with a combination of vemurafenib and cobimetinib. The combination of the dabrafenib and trametinib had actually been further along in developing this biology, in demonstrating that if you give the combination, it should be better than the single agent, dabrafenib, by having better initial responses and more durable responses. It benefits in all of the subgroups of the patients. And then if we look at the development of secondary skin proliferative conditions, it's all improved in the groups where there is the combination of the BRAF and the MEK inhibitor compared to the BRAF inhibitor alone. So what happens when we we're here and we continue to have patients with progression. Well, we still haven't seen a series that tell us what happens when there's progression to BRAF and MEK inhibitor therapy. But many things point to ERK being important. This is the part of the signaling pathway that goes into the cell and turns on a whole bunch of signaling processes. So since I usually think more simplistic than Roger and Mike Davis and many other investigators here in the room, I try to think, well, what's going to be the pharmacological way that we can modify this? And there's been the development by Sherin Plough and also by Biomet Discovery's laboratory of ERK inhibitors that are now in the clinic. So we're intrigued about their potential value, and could we give a BRAF and an ERK inhibitor and get better responses? So this is data from Debbie Wong and Lydia Robert in my lab, where we took a panel of 21 BRAF mutant melanoma cell lines that we established over the years that we have characterized in several ways. And we know which ones respond to vemurafenib and which ones don't. So we know we have a spread of responses. Those are the maroon kind of bars. And we have some cell lines that, despite being BRAF mutant, they're very resistant, innate resistant to the BRAF inhibitor, and some of them are very sensitive. And we tested, would an ERK inhibitor behave differently? So here's just single agent ERK inhibitor, which is in the blue bars, and that's how these cell lines are organized. And there are cell lines that continue to be resistant to inhibition of ERK, suggesting that probably it's not signaling through ERK. And then there's a whole bunch of cell lines that are sensitive, and some of the cell lines that are resistant to the BRAF inhibitor, like these two ones, can be sensitive to blocking further downstream. The Western blots, I'm not going to spend too much time about, but here's a Western blot on a very sensitive cell line to both cells. Here's a Western on a cell line that's partially sensitive to both. Here's one that's resistant to both. And a biomarker of the ERK inhibitor activity would be phosphorylated RSK, and it works actually in all of them, but this one that's resistant probably doesn't have much of an ERK signature and doesn't care if you block ERK or not. This one that's partially sensitive, there is blocking of signaling to ERK. The drug does its effect in the cells that are sensitive, and this one is probably signaling in a different way because we don't have the inhibition of phosphorylation of ERK. So what would happen if we tried to prevent resistance by giving this combination of a BRAF and an ERK inhibitor? So this is an experiment that Lydia and Debbie set up, where we took two cell lines, and I'll show you one and then I'll show you another one, and they were cultured with vemurafenib until the culture would become resistant. They were cultured with the ERK inhibitor until they would become resistant, or cultured with the BRAF and the ERK inhibitor until they become resistant. And this doesn't project too well, but it shows us that already after very few days, the BRAF inhibitor mostly gives us a cell cycle arrest, and there's a whole bunch of cells that are persistent in the culture despite having a major response. But the ERK inhibitors, I'm sorry, it doesn't reproduce here, it really kill a lot more cells up front, at least in the concentrations we used. And at 42 days, the vemurafenib culture was already growing, and the other two were not growing. And we've done this with this cell line and with another cell line, and this is color-coded for growth of cells. So the darker the color means more cells in the culture, and the lighter means less cells in the culture. This is done in 96 well plates, and trying to see how many plates have outgrowth of colonies. And we can see that when there is the ERK inhibitor single agent would prevent resistance compared to the BRAF inhibitor, and the combination of the BRAF and the ERK inhibitor gave a benefit compared to either one. So we've been focusing on this pathway, but we know from what Mike has told us before that there's this other important signaling pathway, the PI3AKT pathway, with this reactivation of receptor tyrosine kinases that happens when you give a BRAF inhibitor that can allow signaling through here, but also a series of genetic alterations that Roger has been describing that I'm not showing here that could activate this pathway. So in sometimes when you have a BRAF inhibitor blocking BRAF, the pathway is not reactivated, and there's this signaling through this other pathway, and we've seen this in many experimental systems. So when we understood a little bit of this biology, then we started thinking, what if we add an AKT and an mTOR inhibitor in cell lines that are resistant both to BRAF and MEK inhibitors? And this was when we did therapies in sequence as opposed to in combination, not because we wanted to, but because we were limited by the materials transfer agreement for which we received the BRAF and the MEK inhibitors. So we could only combine them with tool compounds afterwards. So that was a while ago, but now we want to develop combinations that we can take to the clinic. And this crosstalk between a BRAF mutation being turned off by a BRAF inhibitor and having parallel signaling through PI3 and AKT could be blocked with the new generation AKT inhibitors. And we set up an agreement with GSK to obtain their combinations, because we also had an interest in doing this in the clinic. And actually, we had proposed, this is data that went into a clinical trial that it's open to enrollment now, which is a combination of a BRAF and an AKT inhibitor once there's development of resistance to a BRAF inhibitor-based therapy. So this is data with single agent therapy. Here we have the effects of dabrafenib in our panel of BRAF mutant melanoma cell lines. As I showed before, the same with vemurafenib. Some of them are very resistant, some of them are intermediately sensitive, some of them are exquisitively sensitive. And if we do the same for the AKT inhibitor, we do have more or less the same kind of group, the very resistant, the intermediate resistant, and the very sensitive. We color-coded the cell lines by P10 status, and this is to show that there is P10 null cells that are very sensitive to the BRAF inhibitor alone, and some of them that are very resistant. This is something that we've known for the last 4 years, and many labs have reported on this, where the initial response to a BRAF inhibitor is not hampered by having homozygous or heterozygous deletion or P10. But for the AKT inhibitor, here we color-coded in red the P10 deletions, and in blue one cell line that had an activating mutation AKT. And all of the cells with these alterations are actually in the panel of cells that are very sensitive. Then we put them together. And when we put them together, then we can calculate, do they have a combinatorial effect? And that's what we usually talk about, additive effects and synergy effects. And this can be calculated by something called the combination index, where if you go below 1, it's a positive effect, 1 is additive, and beyond 1 is antagonistic. And we see some cell lines where we don't see much activity. One of them that it's additive, and all of these cell lines where we put the BRAF and the MEK inhibitor as being synergistic. And this includes several cell lines that both Roger's lab and my lab had generated that are acquiredly resistant to the BRAF inhibitors. And to test what we're going to do in the clinic, which is patients who are on BRAF inhibitor-based therapy, who are either BRAF or BRAF+MEK, and it doesn't matter which BRAF or MEK inhibitor. At the time that they progress, we're going to switch them to dabrafenib if they were on vemurafenib, or keep them on dabrafenib, and then add the MEK inhibitor. And if we do this in the cell lines, what happens? In the cell lines that we keep on their cultures, they grow progressively. If we stop the MEK inhibitor and we add the AKT inhibitor, keeping the BRAF inhibitor, we can switch this phenotype, at least in this cell line, and we've done it with all the cell lines. As with the combination with the ERK inhibitor, is it better to do this up front, not just try to chase resistance? And this is what this experiment suggests, which is we give dabrafenib, and we look at how many cells are at day 0, at day 7, or at day 41, we could see that with the dabrafenib, this particular culture develops resistance. We give the dabrafenib and trametinib, we do develop resistance also, but if we give the dabrafenib, trametinib, and the AKT inhibitor, we get a better initial response, and we get a more durable response, and actually we haven't seen outgrowth in these cultures. So I'm not going to go to what's being talked about this afternoon, but I just want to introduce the concept, which is we've been talking about targeting BRAF in the 50% of patients who have the BRAF mutation, but we also know that there's great excitement on the development of inhibitors, or modulators of the immune system, mostly checkpoint inhibitors that take away breaks for the immune system. And if we have these two drugs approved, vemurafenib and ipilimumab, it just made sense to put them together and trying to see if that would be a way of getting better responses and more durable responses. But actually, with this particular combination, it's hard to do. And this is a study we did with Maggie Callahan, who's here in the audience, with Jed Wolchuk and Steve Hody. It was a phase 1 trial of putting ipilimumab and vemurafenib together. We treated 11 patients. After the first 6 patients, we had to go down on the doses because there was liver toxicities. And at patient number 11, we were not allowed to proceed because 6 of the patients developed grade 3 liver toxicities, mostly increases of transaminases, but also 1 patient had an increase in bilirubin, which at this dosing regimen didn't make sense to continue. I still think that something could be done about this. We could do intermittent therapies with the BRAF inhibitor. We could play around with the dose of ipilimumab, things that were not done here. How much time am I? This is the concept, and the concept is that if we give a BRAF inhibitor, we could have a series of effects that modulate the immune system. And most of them will be discussed this afternoon, but it could be a direct effect of increasing antigen presentation. It could be an indirect effect of antigen cross-presentation. It could be an effect not on the tumor, but on the immune system by the BRAF inhibitor, by this phenomenon of paradoxical activation of the MAP kinase pathway. Or it could be that since BRAF is controlling a whole bunch of genes that are giving a growth advantage to this cell, there can be some of these genes maybe encoding for proteins that are immune suppressive in the microenvironment. And maybe that is what we're doing when we block BRAF, then we're taking away these immune suppressive factors and we get a better immune response. So we set up an animal model to test this with a cell line that recapitulates many of the features of human melanoma. It's a BRAF-driven cell line that roses spontaneously on mice generated by Frank Haluska. This cell line has a whole bunch of chromosomal aberrations. This is the chromosomes from several of the chromosomes depicted here with gene amplifications and deletions. We didn't give a carcinogen. This is what the mouse selected to develop this melanoma. And then we set up an adoptive cell transfer model, and the combination improved the effects. And I'm not going to go through that, but mostly on the mechanism, which is we did not see in this mouse model direct antigen presentation. It may be because we're not getting that much of an effect. This is a cell line that has an amplification of BRAF, and it's partially resistant or quite resistant to the BRAF inhibitor. We did not see much evidence of cross-presentation. We did not see direct expansion of T cells, but we saw an activation of the T cells. And this activation of the T cells goes back to the phenomenon that we were talking before that leads to the development of the secondary squamous cell carcinomas, where if we block BRAF in a cell that's driven by BRAF, we have decrease on phospho-ERK. But if we block BRAF in a cell that does not have a BRAF mutation, but instead of that has high signaling properties upstream of it that activate RAS GTP that brings together BRAF and CRAF. If we give the BRAF inhibitor, it transactivates CRAF, and it increases the signaling through the pathway. And we can see this very clearly in this Western blot, where at baseline we have these T cells that are don't have phospho-ERK, and then when we give the BRAF inhibitor, phospho-ERK goes up. Hence, this paradox of doing one thing in BRAF mutant cells and the opposite seen in cells that are RAS GTP activated and are not BRAF mutant. This is a little bit more geeky way to demonstrate this that I learned from Roger's lab, which is you can immunoprecipitate, you can give the BRAF inhibitor, immunoprecipitate CRAF, and then look, does it phosphorylate MEK? And if there was paradoxical activation, there should be an increase in the MEK phosphorylation. This is a kinase assay showing us that it is indeed an enzymatic effect that's induced by giving the BRAF inhibitor, turning on the CRAF inhibitor. This is what happens at the protein level in this pathway, what happens in the signaling level in these cells. Well, this is work that we've done with Tom Graber, where we've taken the T cells, the PMEL cells that are T cells that are specific for a melanoma antigen called GP100. These are CD8 positive T cells that are cultured in vemurafenib. We compare them with squamous cell carcinomas with the gene signature that we obtained from Gideon Bullock that we published in this paper of the paradoxical activation of squamous cell carcinomas. Then we compare it to two bona fide gene signatures of blocking oncogenic BRAF, either the Neil Rosen gene signature or Roger Lowe's gene signature. And we put all of them side by side, and we looked at if we give vemurafenib to BRAF mutant cells, it turns off these genes, it turns off these genes. But if we give them to squamous cell carcinoma, it turns on these genes paradoxically, it turns off some other genes, turns on some other genes. If we give it to the T cells, we can see that it's clear that there's this paradoxical activation because all of these genes are turned on, which should be turned off if we just looked at the effects of the BRAF inhibitor. So we have all of these boxes covered. What about this box? Well, this box is not that easy to study, but we had hypothesized that there may be a change in proteins that are produced by the tumor cells that may interact with the immune system. So to do that, we looked at our gene expression profiling, and I asked Stephen Mock in my lab to rank list all of the genes that were supposed to be the secreted or surface proteins. So genes that would change with the exposure to the BRAF inhibitor that could have a relationship with the environment. And when he gave us the list, and this is just the 108 first genes, then we started seeing that there were a whole bunch of proteins that were differentially expressed whenever we would give this BRAF inhibitor, including CSF1, chemokines, BGF, TGF-beta, and others, all of which were really interesting. There were too many for our group to work on, so we partnered with Mark Smith and Graham MacArthur in Melbourne, Australia, and Mark has accumulated a whole bunch of transgenic mice and antibody reagents where you can block several proteins that you're interested in, give this model of immunotherapy plus BRAF targeted therapy in a mouse. And when he tested our cell line in his mice and tested the monoclonal antibodies that he has been using, there was only one monoclonal antibody that had single agent activity, a monoclonal antibody to 4-1BB or CD137. CTLA-4 and PD-1 did not work here. That doesn't mean it's not going to work in humans, it's just a model. So we just wanted to get an immunotherapy that would work, and whenever he added the BRAF inhibitor, he had this combinatorial effect that was clearly evident. And the combinatorial effect was dependent on the BRAF inhibitor decreasing CCL2 that signals that we could test in CCL2 so the tumor decreases expression of CCL2, it interacts with its receptor, and then we can recapitulate the benefit by doing it in the knockout mouse. So with this, I wanted to touch upon several combinations that can be tested in the lab, can be understood, and then brought into the clinic, including combinations with MEK And this is what I would call a clinically validated combination. Dabrafenib and trametinib has led the way, and cobimetinib and vemurafenib also show similar effects as I've shown you. Combination with the NERK inhibitor, combination with the inhibitors of PI3 kinase or AKT. And I want to highlight this clinical trial that we just activated in SWOG that's taking patients who are progressing on BRAF or BRAF+MEK, and we can add the AKT inhibitor, and then the effects with immunotherapy and how can we understand them. So obviously, this is something that requires a lot of people and quite a bit of funding. So this is acknowledging our funders, and this is acknowledging the people that did the work. So thank you very much.